It's good to see everyone here today. Welcome everyone on Zoom and also those that are joining us at Munich. Welcome to another edition of the special seminar series organized by the McGill Douglas Max Planck Institute of Psychiatry International Collaborative Initiative in Adversity and Mental Health, or AMH for short. My name is Jermaine Lowe and I'm the coordinator for the AMH initiative. And what it is a partnership between McGill University and Max Planck Institute of Psychiatry in Munich, which aims to establish a world-leading research and training program that focuses on the study of early life adversity and how it is biologically embedded and how this increases the risk for psychiatric disorders. So if you would like to learn more about this initiative, please visit our website at www.adversitymentalhealth.com or send me a message at coordinator@adversitymentalhealth.com. Just would like to also let you know that this session is being recorded and the link to the video will be available at a later date. So without further ado, I will pass the mic off to Dr. Naguib Meshawar.
All right. Thank you. Thanks, Jermaine, for your great organizational skills putting this together. So good morning, everyone, and good afternoon at the Max Planck. It's a great pleasure for me to introduce Dr. Martin Teicher. Dr. Teicher obtained his PhD in experimental psychology from Johns Hopkins University and his MD from Yale Medical School, followed by residency training in psychiatry at McLean Hospital and Harvard Medical School. He then took up a faculty position at the Department of Psychiatry at Harvard Medical School, where he has been an associate professor since 1990. He's the director of the Developmental Biopsychiatry Research Program at McLean Hospital, where he and his team study the psychopathologies arising during development, with a strong focus on the lasting neurobiological and psychiatric consequences of childhood maltreatment. His work, which has received uninterrupted NIH funding for the past 30 years, has led to 19 US patents and many awards and distinctions. It has, of course, also led him to publish a great number of highly cited papers. His HN index stands at 86 as of this morning. I was looking at Dr. Teicher's CV this week and noticed that his first 5 first author papers were published in the span of 2 years, with 3 of them in Science and 1 in Nature. And I found that that's a great way to start a career. Since then, he has published nearly 220 more papers, and his elegant work has allowed us to better understand the biological, psychological, and pathological impact of child abuse. In particular, his lab has described using neuroimaging approaches specific regional volume and connectivity changes in the brains of people with a history of child abuse, and hypothesized that these changes are phenotypic adaptations which, during evolution, might have enhanced survival and reproductive success, but may now prove maladaptive. Dr. Teicher's presentation, which will focus on the potential effects of childhood maltreatment on brain structure and function, is titled Childhood Maltreatment, Windows of Vulnerability and Novel Understanding of Resilience and Recovery. Dr. Teicher.
Thank you so much, Professor Meshawar, for inviting me to present. It's a real honor and a privilege to be here, and very excited to be here and to hear about the incredible work that you're all doing. It's just amazing to have such a confluence of people in one place working on the same thing through all sorts of different levels of reduction. I'm sure that's going to continue to have an absolutely astonishing impact on the field. How do I control this one? It's not just me. So, okay, great. Great, okay, thank you. Okay, so let me just start with a definition of childhood maltreatment that I like. I like the World Health Organization definition. It's very broad and inclusive. And it's all forms of physical and or emotional ill treatment, sexual abuse, neglect, or negligent treatment or commercial or other exploitation, resulting in actual or potential harm to the child's health, survival, development, or dignity in the context of a relationship of responsibility, trust, or power. Maltreatment is highly prevalent. About 15% of adults without psychiatric history will endorse a history of moderate to severe maltreatment. You'll find about half of individuals with schizophrenia, bipolar, or major depression endorsing moderate to severe maltreatment, and 3/4 of individuals with adults with persistent depressive disorders will indicate this type of history. It's also a major risk factor. A lot of this we know from the adverse childhood experiences study done by Vincent Felitti and Rob Anda, where they looked at exposure to 10 types of childhood maltreatment or household dysfunction, and they calculated population attributable risk fractions. In other words, if you could magically make childhood adversity go away, how many fewer cases would you have? And you'd reduce anxiety disorders by about 30%, childhood onset psychiatric disorders by nearly half, current depression by about half, and suicide attempts by about 2/3, and about 50% for drug abuse and 2/3 for alcoholism and IV drug abuse. So just think about that. Think about the millions and millions of people, affected by these different disorders and the amount of suffering and pain and cost and damage that these disorders create, and understand that their roots are embedded in childhood adversity. So it's so powerfully important that we understand it and understand it as a biological driver. Another way that they looked at the data prospectively was to see what kind of risk one would have for receiving psychiatric medications in individuals with high levels of exposure. And those individuals who had 5 or more forms of childhood maltreatment, it doubled your risk for being prescribed an anti-anxiety medication, tripled your risk for getting an antidepressant, increased by 10-fold your risk for getting an antipsychotic, and 17-fold a mood stabilizer. So it really goes hand in hand with psychiatric illness. And finally, for those with very high levels of exposure, it was associated with a nearly 20-year reduction in lifespan, and probably due to, major changes in, epigenetics and accelerated aging and telomere shortening and things like that. So what does maltreatment do to the brain? So when I started doing this research, way back when now, like more than 30 years ago, the thinking that we had was that maltreatment was bad for the brain, and particularly bad for the developing brain. And then after a while, it dawned on me that it really makes sense that the mammalian brain, through all the eons of evolution, has not been selected to be relatively resistant to the effects of early stress. And so then the logical alternative is that exposure to early stress generates molecular and neurobiological effects that alter neurodevelopment in a potentially adaptive way that prepares the brain to survive childhood and reproduce in what is predicting to be a malevolent world, but that these changes, which may help you in some extent survive childhood, may be highly maladaptive to the rest of your life. So basically, the first principle is maltreatment does not, in general, result in non-specific brain damage. It's just not randomly, destroying parts of the brain. That there's some very beautiful biology to the kind of things that are affected in terms of its specificity, in terms of its timing, and that they may be understood in terms of their initial functional consequences and then in their maladaptive consequences. So what might be a beneficial phenotypic adaptation? So what might make sense is to be able to enhance your ability to detect and respond to threat if you're living in what you're predicting to be a malevolent world. And this is a circuit diagram for how the brain detects potential threats. You can see that there's 2 pathways. There's basically a low road from the thalamus that pretty much leads directly to the amygdala, and that can generate a very rapid response below the level of consciousness to a stressor or to a threat. And then there's a high road that goes up to sensory cortex that brings the sensation into perceptual awareness, and that can pass through the inferior frontal gyrus dorsolateral prefrontal cortex into prefrontal regions, which can then process the information and get a slower response. They can modify the amygdala's response to threat. And these are all targets potentially for the effects of childhood maltreatment. In other words, looking at them anatomically, we're particularly interested in the thalamus. We're interested in things like the visual cortex, the fusiform gyrus that's looking at facial expression, the amygdala, the hippocampus, key prefrontal regions like dorsal medial, ventral medial, and anterior cingulate. And I'll talk about a fair amount of the pars triangularis. Oops, now it's not working. Oh. Good. Okay. Thank you. Thank you. So what I'd like to do is like to talk about some of these key regions and how they're affected. So let me start with the amygdala. This is a very amygdala-centric circuit. And the amygdala is a key limbic structure that's critically involved in the encoding of implicit emotional memories and is detecting and responding to salient stimuli such as facial expression and potential threats. And structural and functional abnormalities have been observed in the amygdala in a whole host of psychiatric disorders, including PTSD, social phobias, specific phobias, unipolar and bipolar depression, autism, borderline personality disorder, and schizophrenia. The vast majority of the psychiatric disorders have had some evidence for amygdala dysfunction. And we know in terms of translational studies that exposure to stress leads to persistent neuronal hypertrophy and symptoms of anxiety, and that these are very enduring. They don't easily reverse with time. And even when the amygdala is developing and gets more innervated by the prefrontal cortex, that does not cause these changes to revert. And what one generally sees if somebody has a history of childhood maltreatment and you put them in a scanner and you expose them to threatening images, that you'll see increased activation in the amygdala, particularly on the right side of the amygdala. And on the bottom here, your axis is your childhood trauma questionnaire score. And the higher your CTQ score, the greater on average is your amygdala response to threat. So we're particularly interested in the idea of sensitive periods, with the conception that the brain is going to be modified by exposure to stressors, and that the parts of the brain that are going to be particularly susceptible may depend on their development, with brain regions going through rapid changing being particularly susceptible. And some of this work we did in conjunction with Carolyn Lyons-Ruth, who's an attachment researcher at Harvard. And she had a sample of individuals that she followed. She assessed them for their attachment behavior at about 18 months of age, and then followed them for the next 30 years. And we were able to get some funding to bring in a subset of her individuals and to look at their brains and compare them to healthy controls. And what we found was that on average, the more exposure they had to maltreatment, the greater the volume of the amygdala was on the right side. Both the right and left side were equally enhanced, equally enlarged in the individuals who had were part of Carlin's sample. But on the right side, we could see this direct association with maltreatment. And we can see that in particular, it was exposure to maltreatment at around 10 or 11 years of age that was particularly associated with alterations in amygdala volume, in this case, enlargement in amygdala volume. And we didn't see anything on the left side, but when Carlin looked at her attachment data, she found that the volume of the left side correlated significantly with disrupted attachment at 18 months of age. So the more disrupted the attachment, the greater the amygdala volume was 30 years later. Now, it's interesting that we get this differential right or left response. And one way to think about this, perhaps, is that we have a right and left system for detecting threat. And there are two very important types of developmental threat that a child can face. The first type of developmental threat can be a threat to the attachment bond and getting, rejected or abandoned, by the parents. Another kind of threat would be other types of things like childhood maltreatment. And it's conceivable that the left side may be specialized for assessing threats to attachment, whereas the right side may be assessing threats to other types of danger. And that makes sense to some degree in terms if we think about lateralization in the brain. There's a lot of research that suggests that the left hemisphere is specialized for the perception and expression of emotions that lead to approach responses, whereas the right side is specialized for perception and expression of behaviors that lead to withdrawal responses. And if you're detecting a disruption in the attachment bond, approach may be the solution, whereas another kind of threat, withdrawal, might be a more appropriate response. Now, those are the structural changes in the amygdala. How about functional changes in the amygdala? Now, I showed you a slide that showed that the amygdala could be hyper-responsive to threat, and that's what we commonly see with childhood maltreatment. It's also the case that there's a variety of psychiatric disorders in which the amygdala has a blunted response to threat. And these are things like conduct disorder, substance use disorder. You may see a blunted response to threat. And maltreatment is a risk factor for both types of disorders. It's a risk factor for the internalizing disorders like anxiety and depression and PTSD that you see with the hyperactive response. It's a risk factor for conduct disorder, substance use disorder, disinhibited behaviors that you can also see with maltreatment. So the question is there types of maltreatment, timing of maltreatment, that may lead either to a blunted amygdala response or an enhanced amygdala response? And we looked at that in 202 participants that were between 18 and 19 years of age. And we used a scale that we developed along with Angelica Paraga called the Maltreatment and Abuse Chronology of Exposure Scale, which, like the ACE, looked at 10 types of risk, 10 types of adversity. It's somewhat different. We dropped things like parental history of depression or substance use that may be confounded with genetic factors. We added things like peer emotional abuse and peer physical abuse. We have these 10 types of maltreatment, and the instrument collects data on timing of exposure to each one of these classes. And so you can look at this developmentally and see that things like emotional neglect and physical neglect are relatively consistent across childhood, with things like parental physical maltreatment peaks when the children are early and drops off. Things like parental verbal abuse will tend to peak later in the teenage years. Witnessing violence either to parents or to siblings will peak in the middle of childhood. And you can see that sexual abuse shows a strong gender difference, particularly after about age 7, and pure physical bullying also shows a strong gender difference. And a lot of this work has been done with Leptic Khan and Zhenju Zhu, who came over from China to complete his PhD thesis. And we exposed individuals in the scanner to Hirari's emotional face matching task, in which they saw fearful, angry faces, neutral faces, and simple geometric shapes. And what we did was we used artificial intelligence, predictive analytics to identify type and timing that were the most significant risk factors for altered response. And if you look at bold response within the amygdala and compared response to negative faces versus neutral faces, you see that degree of bold response was predicted to a large extent by things like exposure to nonverbal emotional abuse, the NVEA, at about 9 years of age, EN emotional neglect at about 14 years of age, pure emotional abuse, pure E about 17, pure physical at about 6 and 11, and witnessing interparental violence, WIPV, at around age 9. So these were the significant risk factors. And if we put them up in a table, you can see these were the kinds of factors that were associated with the blunted response, and those were the early exposures between age 6 and age 11, whereas those that were associated with enhanced response were forms of emotional abuse or emotional neglect that occurred between age 14 and 18. And if you look at the time course of bold response, you can see on the right the difference in response to the negative face versus the neutral face in the unexposed individuals who didn't experience any of these types of adversity. And you can see there's a somewhat greater response to the negative faces than to the neutral faces. If you look at the middle column, you can see the individuals who had exposure only to these later types of adversity. And you can see a dramatic difference in terms of a much greater bold response to the negative faces versus the neutral faces. Whereas those that had only the early exposure, you can see the exact opposite response. You can see an enhanced response to the neutral face more than the negative face. So you blunted the negative face and you enhance your neutral space response. And we can also compare these groups in terms of their risk for different psychiatric disorders. Major depression is very high and high risk in the individuals who had the later exposures. It's significantly less of a risk in the individuals with just the early exposure. You can also see that obsessive compulsive disorder, for some reason, was enhanced in its risk by those with the early exposure, and that both the early and late exposure increased your risk for personality disorders. So that's the amygdala turning to the hippocampus. It's also a key limbic structure. It's critically involved in the formation and retrieval of explicit memories, including autobiographical memories. It's also affected by basically the same array of psychiatric disorders. We know that stress actually exerts the opposite effect, in a sense, on the hippocampus. Instead of altering the morphometry of the pyramidal cells to have more dendritic branching and boutons and stuff, it actually is leading to shrinkage of the pyramidal cells, and it also suppresses neurogenesis in the dentate gyrus, and these effects are more reversible. And a lot of this work was done by Carl Anderson and Kyoko Ohashi in the lab. And anatomically, what we found is that there was very strong gender differences, and that for males, what seemed to have the greatest effect on hippocampal volume was exposure to neglect during the first 7 years of life, whereas in females, it was exposure to abuse, particularly at ages 10 and 11 and 15 and 16. So strong differences there. We've often seen gender differences. Sometimes males and females have the exact same ages of sensitivity, but may be sensitive to different types of adversity. But in this case, there's a radically different set of potential risk factors. In terms of the function of the hippocampus in this task, in terms of how it responds to threatening versus neutral facial expressions, we found that there's particularly exposure at 9 and 10 that was associated with the blunted response, exposures to emotional neglect and abuse between 14 and 17 that was associated with an enhanced response. And you can again see in the middle panel how that response is enhanced in individuals who had the late only exposure, and is blunted in this case in those who had the early exposure. Also looking at sensory cortex, and we addressed this in terms of the question, does the nature of the maltreatment matter? And this work was done by Jiwook Choi and Akemi Tamada, who were visiting scientists in the lab. And we used fiber tracing and gray matter analysis to look at the effects of maltreatment of different types on aspects of white matter and gray matter, and we used basically blind whole brain analysis or unbiased whole brain analysis, in which basically all of the data is fed into the computer, and the computer indicates those regions that may differentiate between individuals who experienced high levels of parental verbal abuse versus individuals who didn't, or individuals who experienced sexual abuse versus those who didn't. And he went out of our way to select individuals who had very high exposure to one type of maltreatment and only one type of maltreatment. And what we found was that individuals who experienced high levels of parental verbal abuse, that it was associated with alterations in gray matter volume, particularly in the left superior temporal gyrus that corresponds to the auditory cortex. And it was associated with alterations in the integrity of 3 fiber pathways. The one that was affected most is the arcuate fasciculus, and the arcuate fasciculus interconnects Broca and Wernicke's area and is a key language pathway. And the more this pathway was disrupted, the lower the individual's verbal comprehension scores were. In contrast, individuals who had visually witnessed multiple episodes of domestic violence had a selective reduction in gray matter volume in the visual cortex, particularly primary visual cortex. And it affected the integrity of one fiber pathway, which is the inferior longitudinal fasciculus, which interconnects the visual cortex to limbic system and is involved in the memory and emotional response to things that you see. So just think about that. Think about the selectivity with verbal abuse affecting auditory cortex and visually witnessing domestic violence affecting visual cortex. For sexual abuse, we also found alterations in visual cortex, particularly in secondary visual cortex involved in facial recognition. And then Christine Hyman looked at this independently, looked at women with a history of multiple episodes of penetrative sexual abuse. And what she found was selective thinning of the somatosensory cortex and the portion of the somatosensory cortex that's responsible for touch and feeling and sensation from the clitoris and surrounding genital area. So there's highly selective effects of exposure to maltreatment. And we know that sensory systems are our primary filter to the outside world. And we know that they're very plastic and very modified by their environmental experience. In terms of the prefrontal cortex, parts of the prefrontal cortex that are particularly important are the dorsal medial prefrontal cortex, anterior cingulate, and ventral medial prefrontal cortex in terms of regulating response to threat. And the studies are very consistent in terms of showing decreased volume in the anterior cingulate, ventral medial, and dorsolateral prefrontal cortex. In terms of this same basic story that it's basically forms of emotional neglect and emotional abuse that lead to an enhancement in response to these regions. And in this case, neglect, pure physical abuse, or witnessing interparental violence that leads to a blunting of response. Again, this reverses the nature of the response from an enhanced response to negative faces to enhanced response to neutral faces. It's very similar for the ventral medial prefrontal cortex. Again, emotional neglect or emotional abuse enhancing the response during the teenage years, and neglect and pure physical and interparental violence blunting the response. And similar for the inferior frontal gyrus. Now, what are the clinical consequences? McCrory and colleagues proposed that functional abnormalities in threat processing would act as a latent vulnerability that would increase risk for developing anxiety disorders rather than directly producing symptoms. And so we use logistic regression modeling to assess whether differences in regional bold response to negative versus neutral faces would associate with a lifetime history of anxiety disorder. And what we found was that it was bold activity in the ventral medial prefrontal cortex and in the inferior frontal gyrus pars triangularis that predicted their risk for anxiety disorders, and that basically an increase in ventral medial prefrontal response was associated with a markedly reduced risk for anxiety disorders, where an enhanced response in the inferior frontal gyrus was associated with a markedly increased risk, basically a five-fold increased risk for anxiety disorders. So maltreatment exerts prominent effects on key components of the threat detection and response system, which can lead to a blunted response or can lead to enhanced response, depending on the timing of exposure. And sensory systems, which are a primary window to the external environment, are selectively modified by types of maltreatment that they convey. And there are prominent effects of maltreatment on the reward system, which I wouldn't have time to get into. In general, maltreatment is associated with a blunted striatal response to anticipation or receipt of reward. Maltreatment is also associated with marked effects on white matter tracts, corpus callosum in particular. And the corpus callosum is the largest white matter tract that interconnects the right and left hemispheres. And they may be, particularly in the genu, affecting hemispheric integration. And that, interestingly, we find, again, gender differences. We find in females that the myelinization is particularly affected by maltreatment, whereas in males, it seems to affect the integrity of the axon and axonal infrastructure to a greater degree. So this was a focus particularly on independent regions of the brain. We've also looked at this more at the network level. And this work has been done by Kyoko Ohashi. And the big question that we're trying to understand is, why are some individuals who've experienced rather substantial amounts of maltreatment resilient? We know that it's a major risk factor for depression and anxiety, and from your work, suicides and other psychiatric disorders like substance abuse, but there's some individuals who sustained a great deal of maltreatment who seem to be doing remarkably well or much better than expected. And so we first asked the question, are the brains of individuals with history of maltreatment but without psychiatric symptoms unaffected? And we figured, well, maybe psychiatric and neurological alterations would go hand in hand. And the brains of individuals that were resilient would look pretty much like unexposed controls, where the brains of the individuals who had maltreated and had psychiatric problems would show all of these abnormalities. And we were completely and absolutely wrong. These are brain changes that have been reported in individuals with a history of maltreatment who have no psychiatric symptomatology. And you actually see in this group basically the exact same constellation of changes in stress-susceptible brain structure that you see in maltreated individuals with severe psychopathology. It includes things like changes in the cingulate, the prefrontal cortex, the insula, amygdala response to threat, white matter tracts. All of these things are affected in individuals without psychiatric illness. So, it's really pretty stunning and sort of hard to believe for a while. This suggests that these psychiatrically resilient maltreated individuals are not neurobiologically unaffected, rather they are effectively compensating through some other mechanisms. And the critical question is, how do they manage neurobiologically to maintain mental health despite a host of abnormalities in stress-susceptible structures? So we took our sample. We had 342 individuals that we had good maltreatment data on and neuroimaging on, and we're able to divide them into three groups. We had a control group that had very low exposure to maltreatment, and we had two maltreatment groups that were matched as close as we could for the degree of exposure to each one of the 10 types of maltreatment. Didn't differ in any of them significantly. And one of the maltreatment groups was highly symptomatic, and the other group of maltreated individuals didn't differ significantly from the controls in the degree of psychiatric symptomatology. So we had a maltreated asymptomatic group and a maltreated symptomatic group, and we had an unexposed control group. And we figured that if we're going to understand what's going on across all of these brain regions that are connected, that we'll have to use something sophisticated like network analysis to look at the interconnections between these regions and how they operate together. So just as a brief primer, network analysis has its origins in social psychology and sociology with the study of social networks. In a network, each individual is a node, also called a vertex, and the lines connecting individuals to each other are edges or links. The number of edges associated with a node is its degree of centrality. Typically, some individuals have more edges than others, and a hub is a node with a much greater number of connections. And you can see in this simple social network that Diane has the most connections. She would have the highest degree of centrality, and she would be a primary hub within this network. Social networks like brain networks and computer networks often share an important property called small worldedness. And this is what a small world network would look like. You would see in a small world network that you have communities, and in these communities you have a lot of interconnections between the brain regions within each community. They would be highly interconnected, and then you have somewhat sparser degree of interconnection between the communities. So you have to have this balance of segregation and integration. And and this is sort of an optimal design for things like brain networks, computer networks, social networks, because it allows effective communication between all of the nodes in the network, but it isn't anywhere as near as demanding as trying to interconnect every other node to every other, because these connections are metabolically expensive. So this is a sort of an optimal configuration. And the average number of steps that it takes for each node in the network to connect to every other node is the average path length. The global efficiency of a network is the inverse of the average path length. And the degree of small worldedness reflects the ratio of local clustering to average path length. Another property of networks is their vulnerability. And one way to calculate it is to assess how much the removal of a node from the network affects the global efficiency of the network. The network's vulnerability is then defined as the consequence of removing the node with the greatest impact. And so if we look at that, we look at the unexposed controls and the symptomatic and asymptomatic maltreated network individuals. Again, we see the same thing. We see that the symptomatic and asymptomatic maltreated individuals have the same alterations in network architecture. There's a reduction in global efficiency, there's an increase in small worldedness, and there's an increase in vulnerability. And there's really no difference between the two groups. So what we thought is that resilience may occur in individuals with vulnerable networks abnormally functioning, may result from the diminishing the impact of potentially problematic nodes. So if you have a node shown in red that's like misfiring or overly sensitive, and you had fewer connections of that node, that may enable an individual to effectively compensate. So if you just turn down the volume slightly in a problematic node, would that allow for an effective compensation? So I looked at that particularly in terms of the right amygdala, which tends to be hyperreactive in individuals with maltreatment. And we compared the three groups, and we're using a measure of nodal efficiency, which is it's the measure of the importance of a node, its ability to spread information to other nodes in the network. And what we found, looking at the right-hand side, was that the nodal efficiency of the right amygdala was the same in the controls as in the maltreated susceptible group, but it was reduced in the maltreated resilient group. So that was one difference between these groups. And then we used machine learning to identify whether there are other nodes in the network that differed in nodal efficiency. Now, they could have differed in terms of, could have had increased nodal efficiency, they could have had decreased nodal efficiency. We identified 9 nodes, including the amygdala, and all of them had reduced nodal efficiency in the resilient group. Now, it may seem confusing that reduced nodal efficiency was associated with resilience. The normal inclination would believe that resilience should stem from enhanced connectivity. However, when it comes to the brain, bigger may not be better and less may be more. And postnatal brain development goes through both an overproduction and pruning phase with marked increments in efficiency and performance, resulting in the strategic pruning of connections. And in network design, it's not beneficial for nodes to have as many connections as possible. They need to have an optimal number and configuration of connections. And in what's a sparser network in the maltreated individuals with fewer frontal hubs, it may well be optimal for certain nodes to have reduced connectivity or nodal efficiency. And you can understand these findings if you're thinking about it in terms of turning down potentially problematic regions. And so what do we know about these potential resilience nodes? Well, I only have time to talk about one of them, but I'm going to get back and talk about the inferior frontal gyrus pars triangularis. And you can see in this graph here showing the interaction between maltreatment and depression, that individuals with high nodal efficiency in this region show a very dramatic relationship between their degree of exposure to maltreatment and their degree of depression. Whereas individuals on the left who have low nodal efficiency in that region show like no relationship between maltreatment and risk for depression. They're resilient to depression. Same thing on the bottom. Those with high nodal efficiency are very susceptible to anxiety. Those with low nodal efficiency are not. And the pars triangularis contains a portion of Broca's area and seems to play a role in voices inside your head. And it may be responsible for the self-castigating voices that maltreated individuals often perceive. And reducing the influence of this region on the overall network may enable maltreated individuals to more effectively compensate. Bonanno has done some very nice work looking at trajectories of individuals exposed to adversity. And you can have 3 basic trajectories. You can have a minimal impact trajectory in which you become slightly symptomatic, but then go quickly back to baseline. You can have a trajectory where you become highly symptomatic, but then over the course of time recover. And you can have a trajectory where you become highly symptomatic and stay chronically ill. And the question is, what's going on with these resilience nodes in the individuals who have this minimal impact versus the recovery pattern? And basically, the individuals at the minimal impact pattern have reduced nodal efficiencies essentially throughout, these regions, whereas those recovered have reduced nodal efficiency in only 2 regions, which was the right amygdala and the right subcallosal gyrus. So it may be the case that treating an individual with a maltreatment history doesn't require reversing the effects of childhood maltreatment. It may be fostering some compensatory changes that move the brain architecture from the susceptible more into the resilient configuration. And that may enable recovery, and that we may be able to design targeted treatments that just do this type of change that enhance this aspect of network architecture to enable them to effectively compensate. Last thing I'd like to mention is something I've been working on with Jackie Sampson, which is the ecophenotype hypothesis. And that what you can see is that for many psychiatric disorders, that there is a high prevalence of individuals who have a history of childhood maltreatment. And there's also a significant number of individuals with the same DSM or ICD disorder who do not have a history of maltreatment. And that for many of these things, it may be close to 50-50 in terms of who has a psychiatric history and who does not have this history of maltreatment. And what we propose is that maltreated and non-maltreated individuals with the same primary diagnosis are clinically, neurobiologically, and genetically distinct. The ones that have the maltreated version have an earlier onset, they have a more severe course, they have more comorbidities, they have greater symptom severity, and they have generally a poor response to treatment. This is one example of that. This is looking at individuals through the iSpot-D study where they were prescribed a first-line antidepressant for acute depression. And this is prescribed, in this case, one of 3 common selective serotonin reuptake inhibitors, and looking at the percent of individuals who showed a remission. And that you had 84% of the individuals with no history of maltreatment showing a remission, and only about 16% of those with the maltreatment history, particularly with maltreatment between ages, I think, 4 to 7. So maltreatment can markedly reduce your response to first-line treatments. There also appear to be autoimmune, metabolic, cardiovascular, inflammatory alterations in the maltreatment group, and we also have data showing some marked risk factor also for obesity in psychiatric patients. Also, the hippocampal and amygdala differences that are often reported in depression may be specific to the maltreated subtype. And basically, there's about 9 different psychiatric disorders associated with reduced hippocampal volume. And in all of those, childhood maltreatment is a risk factor for all those things. It is basically the driving force. And what we wind up thinking is that in the maltreated subtype, you're going to see morphological abnormalities, particularly in stress susceptible structures, which you'll often not see in the non-maltreated subtype. And in the non-maltreated subtype, we're probably dealing with neurochemical or functional abnormalities that aren't showing up in terms of morphometry. And so I think that's the a really interesting hypothesis that we're pursuing. So maltreated and non-maltreated is the same primary diagnosis, but clinically neurobiologically distinct. And this has far-reaching implications. We really confound a lot of the research that we do if we combine these individuals in the same group, because their biology may be vastly different. And so that may lead in part to the field finding results that don't readily replicate. Combining them in the same group may also distort findings regarding the efficacy of treatment, because the treatments may be remarkably effective in one subtype and hardly effective at all in the other subtype. And so distinguishing them based on this factor may really help, I think, advance progress in the field and lead to more consistent findings. What we're currently working on are substance abuse ecophenotypes with the idea that there's 2 driving factors for substance abuse. One, of course, is maltreatment. And in the non-maltreated individuals, what seems to be a big driving factor is attention deficit hyperactivity disorder. You can also see a lot of ADHD in the maltreated subtype, but that's not a big determinant in that group. The maltreatment is. But in the non-maltreated group, the ADHD then takes over as a major risk factor. We're also working on an instrument that's compatible with the MACE that will look at the chronology of perceived discrimination, so we can start to understand how much discrimination at different stages of development is a risk factor. And we've been looking at the effects of various forms of treatment, We've been looking at mindfulness and published a series of studies showing that it does have effects on amygdala volume and hippocampal volume. We're starting a course of studies on individuals getting dialectic behavior therapy, and we'll be looking at transcranial photobiomodulation as a stimulatory type of treatment that may also have effects and benefits for these individuals. So I also want to thank out to Cindy McGreenery and Elizabeth Bolger, who collected a lot of this data, and thank you for the opportunity to talk.
Thank you. Yeah, can you hear me? Oh, okay. Thank you, Dr. Teicher, for a very nice talk, a great overview of your work, your contributions to the field. Let's open the floor to questions. Are there any questions?
Thank you very much. That was excellent, an excellent overview of very impactful and work that you have conducted over the years. I was wondering if you could comment a little bit on the timing of the abuse. So you showed data that clearly modifies the responses both in the amygdala and the hippocampus, depending on when the abuse took place. So I was wondering if you could elaborate a little bit more on what you think is behind, and how is that responses are so markedly different depending on when the abuse occurs.
Yes, it's a wonderful question. It's the big problem, trying to understand that. So I think on a simple level, we're thinking about this in terms of sensitive period, and the idea that brain regions become plastic, particularly during stages of development when excitatory and inhibitory influences come into balance, and the window of responsiveness to the environment opens up, and then this may be closed by molecular breaks and perineural nets. And so there may be a transient period in which the brain is particularly responsive. It may be more complicated than that. One of the things that we found, for instance, and goes along to a significant extent with the human literature, is that if you look at childhood abuse and the hippocampus, you can see the hippocampal effects pretty clearly in adulthood. But if you're looking at children themselves, most studies find no difference. And we modeled that in rats with Sue Anderson, and basically found that stress would lead to alterations in synaptic density in the hippocampus if you looked at them at a particular age. But if you looked at them at a younger age, you wouldn't see it. And so it may not be modifying the hippocampus itself. It may be producing epigenetic changes or other kinds of alterations in molecules that are involved with hippocampal development, and that those factors may have this window in which they're vulnerable, and that they may then show effects on growth at later stages of development. So I think it's a complicated question that we don't have a full understanding of. Do you have some thoughts?
It really depends, I think, on the development,
Yeah.
and then also on the type, I guess, on the type of abuse,
Yeah.
and the type of maltreatment. But I don't, and I think that that's an important, question to clarify.
Yeah, and I think at this stage we can point fingers at what seems to be some of the types and what seem to be the windows, which will require replication. And then I think the more molecular scientists hopefully can pull apart the answers, perhaps some neuroautopsy studies. The interesting thing is, one of the main theories that's currently in vogue is that you can think about adversity in terms of two types. You can think about adversity in terms of deprivation, and you can think about adversity in terms of threat. and that things like physical and sexual abuse would be threat, and that things like emotional or physical neglect would be deprivation, and low financial resources would be deprivation. Our findings cut across that you can see both neglect as a associated with a blunted response and neglect associated with an enhanced response. And you know, the McLaughlin-Sheridan model basically leaves out emotional abuse, and they focus more on physical sexual abuse, but it's really emotional abuse that affects the timing within this part of the circuit as opposed to physical and sexual. We find other things are markedly affected by physical and sexual abuse, but in this case, these things seem to be more affected by emotional abuse. So I think that can't neglect emotional abuse. It's particularly for psychiatric problems, it's a huge risk factor.
I agree with that. In that sense, did you, in the studies, did you separate the deprivation, maltreatment more related to deprivation versus maltreatment more related to threat, or did you include them together?
Oh, no, we looked at each of the 10 types of maltreatment. So we had, for instance, okay. So here we have the 10 types of maltreatment, and you can see here that physical neglect and emotional neglect are involved, but so is pure physical abuse, witnessing interparental violence. And then for the enhanced response, you can also see emotional neglect, a lot of parental verbal abuse, and pure emotional abuse are risk factors. So we looked at 10 types of maltreatment. We had enough of a sample size to be able to do this if we pooled males and females. It's nicer to do that with the larger samples. And we've also now looked at it in terms of emotional threat, physical threat, emotional neglect, physical neglect as risk factors. It makes a huge difference. For instance, if you do that in terms of psychiatric symptomatology, you can see that for depression and anxiety, it's really emotional threat that drives it. Where if you're looking at PTSD, it's physical and sexual threat, that drives it. So that, what's optimal in terms of looking at, in terms of different types of maltreatment is unclear. I think 10 may be a bit overkill, but 2 may be insufficient.
Thank you. Thank you. Very nice talk and very nice overview. So it's very nice and very interesting what you say about resilience and susceptible individuals. And I guess like you're assessing mainly like the long-term changes. I wonder if there would be differences, or what do you think about more immediate responses that would actually influence resilience or susceptibility?
Right. So, that's a big question in terms of with your, level of reduction, level of understanding what's going on. Are there particular symptoms that occur early that would predict that you might have, a chronic course versus a recovery course, or that's going to predict that you'll have a minimal impact pattern versus a chronic pattern? And then it's also, are there protective factors, or are there other factors that may modify the course? We're thinking that some of the individuals, particularly those who have the minimal impact pattern, may start off with these brain differences, and they may just be not, more in a cannulated course where they're going on their trajectory and not impacted by this exposure. Whereas the ones who show the recovery pattern don't have these differences to begin with, and that they emerge over the course of time as they recover. But it's a great question. What we have looked at in terms of trying to understand that is whether they got ill enough to have received a psychiatric diagnosis or to have had a lifetime history. So you also have a second, definition where we've compared individuals who've never met diagnostic criteria for psychiatric illness versus individuals who have met diagnostic criteria. And we see the same basic story if you define it that way. But this data that we have is retrospective data. We're looking at individuals right outside of adulthood, right outside of childhood, so they can have a pretty clear memory of what happened in their childhood, but they're 19, 20 years of age when time we scan them. It would be very nice to have prospective longitudinal data. I think it's hard to get that level of detail about maltreatment history. It's a real shame that in something like the ABCD study that they're not getting really, really good data on adversity. I mean, they're getting some data on adversity, but not all of the things one would really like to know. But you'd really, really want to have a prospective longitudinal study, to really see that. And you do really want to know, what may be going on, symptomatically that may lead. And the big question is there something that you can pick up early that would be a target for treatment? That would then be something that you would use. And so we have a hypothesis. The symptom complex that we see the most with maltreated individuals is something that we call limbic irritability. And that these are symptoms that are the kind of symptoms that individuals have when they have temporal lobe epilepsy. And these are things like deja vu and jamais vu kind of phenomenon, their perceptual distortions, their hallucinatory events, their disturbances, and their automatisms and things like that. And if you look at symptoms of maltreated individuals, things like depression or dissociation or anger or anxiety, limbic irritability is most increased and most significantly correlated with maltreatment. And then we find in our study, it's the biggest risk factor for substance abuse prospectively. So their level at 18, 19 years of age would really predict their risk for substance abuse down the road. And the question is, if that's going on, it may be affecting maltreatment's effect on the limbic system, can you try to target that? Can you try to treat that? And may that have a preventative effect if you did? So we're hoping to get some funding to do that.
We'll take one question from Zoom and then two more from the audience.
Sure.
Okay, so this question, everyone hear me?
You want me to read it?
This question is actually from Shangfei Meng. Thank you for the comprehensive presentation in the area of maltreatment and neurobiological changes. Could you comment on the measurements of maltreatment as well as resilience that are used in your studies?
Okay. So yeah, I passed over these things quickly. Yeah. So for maltreatment, we use a scale that we created called the MACE, the Maltreatment and Abuse Chronology of Exposure Scale. And this is basically a 52-item scale that asks individuals if they experienced certain events in their life. And if they did, they then check off throughout their life which years of occurrence that they had. So, and we used item response theory to break them into these 10 categories of maltreatment. And the scale has some very nice properties. It has a very high test-retest reliability, about 0.91, and that even within each type of abuse, there's high reliability. And if you look at the arc over the course of age, there's high reliability. So the scale is very reliable. That's true for a lot of other maltreatment instruments. They're usually in the 0.88 to 0.91 range in terms of the test-retest reliability. We don't know what the validity is. We do have some inklings that in looking at the individuals who are part of Carlin's, Lyon, Ruth's longitudinal study, that those who experienced problems with attachment on the MACE, they had significant problems at around age 2 with emotional neglect and not any other area. So that was nice, confirmation. There's also a paper from the Avon longitudinal study identifying brain regions that were affected by exposure to adversity during the first 6 years of life. We can look at the same brain regions on the MACE and show that, yes, indeed, those brain regions had sensitive period during the first 6 years of life. And a lot of the other brain regions that they didn't see affected don't have sensitive periods during those time. So we have some confluence of information that suggests that the scale is useful. This was published in PLOS ONE in 2015, and I made it publicly available, open access, free to use. The scales available online and the scoring for the scales available online. If you're looking for, RevCap program or something like that, happy to provide it for you if you want to include it in your work.
Thank you very much for this great presentation. I wanted to come back to you showing the nodes of resilience or this concept of resilience with the network. And first of all, that was based on functional connectivity, or was it based on morphological data?
Based on
both?
structural connectivity.
On what?
Structural connectivity.
Structural connectivity. And so what would be your approach to, say, toning down the volume? How would you approach this in terms of fostering resilience? If we can do something about it.
Right. So let's assume that the inferior frontal gyrus pars triangularis is one of these nodes. And let's assume that we know that it plays a significant role in the inner voices in your head. It's part of Broca's area, it's a speech center, and it also seems to be responsible for the voices in your head. And let's assume that in maltreated individuals, that what it's saying is really not nice. That it's saying, you're fat, you're stupid, you're ugly, you're never gonna amount to anything, and I wish you were never born. And that what we see is that this connects, and it particularly connects to things like your default mode. And that we had an individual in our group develop a treatment targeted for individuals who experienced high levels of parental verbal abuse. And by essentially exposing them and diminishing their response to the things that they heard from their parents, and they got less symptomatic. And what that was correlated with was reduced connectivity, his functional connectivity between the pars triangularis and the default mode. So that reducing connectivity, reducing influence may enable you to more effectively compensate. Just by, if you're compensating for something that's a problem, if it's a bit less of a problem, you may be more able to functionally compensate. So that's our thinking. It isn't any more sophisticated than that. That's the point we're at in terms of thinking about it.
Thank you. We are almost running late, so I'm just going to ask the last question, if you don't mind. Could you tell us a bit more about transcranial photobiostimulation, and if this method is a strategy to reconfigure the brain towards a resilient phenotype?
Sure. So this is work that Fred Schiffer is pursuing in the lab. And Fred's been interested for a very, very long time in right or left hemisphere. And with the idea initially that maybe some of the memories of trauma were embedded in the right hemisphere, unavailable to conscious recall, but they could jump across and become, disturbing and produce flashbacks and things like that. And so we examined that and found that an individual with maltreatment, looking at response to emotionally stimulative versus neutral stimuli, that you activated left and right hemisphere to very similar degrees, and individuals who were normal, those who had maltreatment, tended to activate right during the emotional things and tended to overactivate left in terms of neutral thoughts, and that led him to think about the two hemispheres and whether he can stimulate the two hemispheres. And he wanted a non-invasive way of stimulating a hemisphere. And it turns out that that near-infrared light passes through biological tissue very well. If you held a flashlight to your hand, you can see this dark red color. That's the near-infrared light penetrating through. And near-infrared light gets through your skull. And so he uses a near-infrared laser with a fan that cools it down and holds it over basically prefrontal cortex for about 4 minutes. And this has effects on NADP and cellular activation, and it enhances neurotrophic factors. And there's like 10,000 papers on this aspect of near-infrared light and what it can do to, to biological systems. And he did the first study looking at this in depression. His hypothesis is that there's differences in hemispheric emotional valence between left and right hemisphere, and that basically individuals have. a healthier and more mature personality on one side than the other side. And that this difference may be fostered to a great deal by maltreatment, and that individuals who are highly symptomatic are having their less healthy side more in the forefront. And that if you stimulate with the transcranial near infrared light to the healthier hemisphere, you can make it come into the forefront more. And so we found that it very quickly reversed depression symptoms doing this treatment, and it lasted for a couple of weeks after a single treatment. And he's been using it in his practice now for many, many years, very effectively. And we did this in a phase 1 randomized control trial in an SBIR grant for opioid users, and found that stimulating the healthier hemisphere very significantly reduced craving for opioids. And we're just about to start a larger study, a double randomized control trial, seeing if it actually reduces degree of opioid use in opioid users. But it also has very nice effects on mood and anxiety to stimulate the healthier hemisphere. So it's interesting. It's really simple.
And no side effects?
What?
No side effects?
The individuals don't particularly report any side effects. I tried it. I didn't like how it felt, but I wasn't feeling anything significant. It's sort of just, I felt like things were different. Initially they were brighter, but I didn't feel like I was thinking quite as clearly. And one of my colleagues who also wasn't having any symptoms, she tried it, and she found that she had a hard time sleeping for the next couple of days, that she was like too stimulated. So I think it works well if you have a good reason to use it. I'm not sure if it's helpful if you don't have a reason to use it.
Thank you. The conversation will continue during your student lunch and the meetings this afternoon. So I thank you once more for your great talk. Thank you.
Thank you.